Hello everyone, this presentation is brought to you by Group 3. Today we will talk about monoclonal antibodies. First, let me explain the definition of a monoclonal antibody. A monoclonal antibody is produced from a single B-cell clone, so it recognizes only one specific epitope. Because of this, monoclonal antibodies have extremely high specificity. In contrast, polyclonal antibodies are produced from multiple B-cell clones and therefore recognize multiple epitopes. This difference in specificity is one of the main reasons monoclonal antibodies are widely used in research and medicine. Next, I will explain the classic method for producing monoclonal antibodies, hybridoma technology. It consists of 6 major steps. First, a mouse is immunized with the target antigen. This stimulates B cells that produce antibodies against the antigen. After immunization, we harvest antibody-producing B cells from the mouse spleen. However, these cells cannot survive long in culture. To overcome this limitation, the B cells are fused with immortal myeloma cells. The resulting hybridoma cells combine both abilities, producing the desired antibody and proliferating indefinitely. The fused cells are then cultured in HAT medium. Only hybridoma cells survive under these conditions, while unfused myeloma cells die and unfused B cells naturally disappear. Next, ELISA is used to identify hybridoma clones that produce the specific antibody we want. Finally, the selected hybridoma clone is expanded to produce large amounts of monoclonal antibodies. Although this method is reliable, it has several limitations. Antibodies are mouse-derived, so they may cause immune reactions in humans. Some antigens do not elicit strong immune responses in mice. Also, it is difficult to introduce detailed structural modifications. In addition, the overall process takes several months. These limitations led to the development of more advanced technologies. The first modern method is patch display. Antibody fragments are displayed on bacteriophages, allowing rapid and efficient screening for high-affinity human antibodies. This approach avoids the need for animal immunization. Another major advance is the use of transgenic mice whose immunoglobulin genes are replaced with human ones. These mice produce antibodies that are much more human-like, reducing immunogenicity in patients. A well-known clinical application is nivolumab, an anti-PD-1 antibody. Normally, PD-1 on T cells binds PD-L1 on cancer cells and suppresses immune activity. Nivolumab blocks this interaction, allowing T cells to attack cancer cells more effectively. Monoclonal antibodies can also trigger antibody-dependent cellular cytotoxicity, ADCC. After antibodies bind to cancer cells, NK cells recognize the antibody's Fc region via Fc gamma receptors and release cytotoxic molecules such as perforin and granzyme to kill the target cell. Let me summarize our presentation. Monoclonal antibodies are highly specific and powerful tools. The hybridoma technique established the foundation for antibody production. Modern methods such as patch display and transgenic mice enable the creation of safer, more effective therapeutic antibodies. These technologies play crucial roles in cancer immunotherapy and many other medical fields. Thank you for listening.